This video is talking about cellular respiration. To start off, we have glycolysis, where glucose first gets converted into G6P with the help of hexokinase, and we have the use of 1 ATP. Then G6P is converted into F6P with the help of the enzyme PGI. Then F6P is converted into FBP with the help of the enzyme PFK, and this is where we have a second ATP being used. Then we have FBP being converted into either GAP or DHAP with the help of enzyme adolase. Ultimately, DHAP will have to convert into GAP in order to continue, and that is the end of phase 1. Phase 2 begins with GAP being converted into 1,3-BPG with the help of the enzyme GAPDH. Then 1,3-BPG is converted into 3-PG with the help of the enzyme PGK. And here we have 1 ATP being produced for each molecule. It's also worth to note that 1 NADH for the previous reaction is produced 1 for each molecule. Continuing on, we have 3-PG being converted into 2-PG with the help of enzyme PGM. 2-PG is converted into PEP with the help of enzyme enolase. And finally, we have PEP being converted into pyruvate with the help of pyruvate kinase. Ultimately, we have phase 1, which is the investment phase. 2 ATP are used. Phase 2 is the payoff phase, where there's a generation of 4 ATP, all in all resulting in 2 ATP, 2 NADH, and 2 pyruvate. Next up, we have the pyruvate dehydrogenase complex, where pyruvate is put into E1 and TPP is attached onto it. It eventually gets cycled over to E2, where CoA comes and creates acetyl-CoA, and then we get the resulting molecule that is not attached to acetyl-CoA goes into E3, where 1 NADH is produced for each pyruvate. So ultimately we get 2 acetyl-CoA and 2 NADH. Next up, we have the citric acid cycle, where we have the acetyl-CoA coming in and reacting with the oxaloacetate, and with the citrate synthase enzyme, it results in citrate. Then citrate is converted into isocitrate with the enzyme aconitase. Then the isocitrate is generated into alpha-ketoglutarate with the enzyme of isocitrate dehydrogenase. And in that reaction as well, you have the production of NADH. Then the alpha-ketoglutarate is converted into succinyl-CoA with the help of the alpha-glutarate dehydrogenase enzyme, where another NADH is produced. Then the succinyl-CoA is converted into succinate via the succinyl-CoA synthetase, and this is where 1 GTP is produced. Succinate then is converted into fumarate via the help of the succinate dehydrogenase enzyme, and here we have the production of FADH2 as well. Then fumarate is converted into L-malate, where the enzyme fumarase helps, and then the L-malate is converted into oxaloacetate with the help of malate dehydrogenase, and this is where another NADH is formed. Ultimately, with 1 molecule of glucose, you'll get the production of 6 NADH, 2 FADH2, and 2 GTP. Finally, we have the electron transport chain, where the main part of this is taking the NADH and the FADH2 and generating the ATP by creating a proton gradient across the membrane. This is done using the 4 different complexes below, and complex 1 takes NADH and pushes through 4 protons, and then as you can see, complex 2 actually connects with the citric acid cycle, where the succinate is converted into fumarate, which is when the production of FADH2 is done. And as you can see, there is no protons that is pushed through. Complex 3 ends up pushing through 4 protons, and then complex 4 pushes through another 2 protons. Altogether, with the different reactions that will result in ATP being generated, and also when ATP is used, you end up with about 32 ATP from one glucose molecule. And as you can see, there are different conversion factors. So, for example, NADH actually generates about 2.5 ATP, while FADH2 converts into about 1.5 ATP.